Welcome to the Endpoints podcast, a brand new podcast from the ALS Therapy Development Institute, where we talk to people in the ALS community, from those living with ALS and their families to ALS researchers and fundraisers. My name is Emer, and today I'm joined by Dr. Steve Perrin, CEO at ALS TDI, and Dr. Fernando Vieira, CSO at ALS TDI. And we're going to talk a little bit about antisense oligonucleotides, or ASOs, which are kind of a promising area in clinical development at the moment. We're seeing a lot of promise with approved treatments for SMA, spinal muscular atrophy, and Duchenne muscular dystrophy, and dozens more treatments in clinical trials, including 2 for ALS, BIIB078 in adults with C9orf, and BIIB067 in adults with SOD1 genetic mutations, 2 of the most common heritable genes in ALS. So welcome, Steve and Fernando.
Thank you, Emer. Glad to be here.
So let's start with the most basic question. What are ASOs?
So I'll start by saying I'm certainly not the world's expert in ASOs, but ALS TDI, we do work with them quite often here, both in cell-based models and in animal models. And what ASOs are antisense oligonucleotides. And what that is a molecule, mostly DNA, that is specifically designed to target and suppress expression of a specific gene. They often work by binding right to an RNA molecule that would ultimately produce a specific protein. Sometimes those RNAs just get blocked so that they can't get translated into a protein, and sometimes the RNAs end up getting degraded or chewed up by an enzyme in the cell called RNase H. ASOs are really exciting because it gives you the opportunity to tackle specific types of disease where you know what the exact cause is. So monogenic disorders, like Huntington's is another case. Some versions of ALS, like SOD1, C9orf72, can be addressed with antisense technologies.
So how have ASOs impacted the medical world so far?
So it's a really good question, and as Fernando pointed out, I think for the task of this podcast, I can't go into an awful lot of detail because it would take too long, but it's amazing to me when you think about science how technologies that we take for granted today would have appeared mythical 10 or 20 years ago. And ASOs, I think, is a perfect example of this. If somebody had told me when I was finishing up my graduate degree 30 years ago that you'd be able to take little pieces of DNA and actually modulate genetically and functionally specific genes in the genome, I would have laughed. But yet, when they were first developed 30 years ago, initially in cell lines, that was a really amazing scientific breakthrough. And we all, as scientists, immediately understood the implications of what that might mean, but to have a technology work in a cell growing on a dish was one thing, and I think a lot of us were skeptical about whether you could ever translate that really exciting finding into animals and then into man, and lo and behold, it took a while, but it was an amazing and profound impact when they first started moving ASOs into animal models like mice, and lo and behold, you could actually modulate the expression of specific genes. And it's taken decades to get us there, but now, as you'll hear, the technology is actually working in the clinic. And, we take it for granted today, but those were amazing milestones that occurred to enable this technology platform.
Yeah, and you touched on it a little bit there. So they were discovered about 30 years ago. What is taking so long? What are some of the hurdles that this technology has faced over the past 35 years?
I think there was lots of questions about the specificity, how to deliver them, to improve the stability, to understand, especially as we moved into animals and subsequently into mammals and people, how do you control where they go, how do you administer them, how do you safely monitor for off-target effects? I think most of the gap in the 20 to 30 years was more about developing the technology and improving the backbone of the ASO. It was mostly technology development.
Yeah, I think one of the things that happened with ASOs is that people were trying to use them a lot like your typical small molecule drug. People imagined that you needed to dose a lot and very frequently. And over time, and with modifications to the technologies, like Steve described, sort of advances in the chemistry backbones of these molecules, it was identified that in a lot of cases you really only have to deliver it maybe one time a month, one time every 2 months, to get a long-lasting suppression of your target gene. But that wasn't immediately obvious, and I don't think we have a full understanding yet of why that's the case, why the effects last so long.
Yeah, and I mean, on top of that, we have to give some credit here to Ionis. I mean, when we talk about technology development, the initial first-generation ASOs really lack stability. I mean, cells are designed to get rid of small pieces of RNA and DNA, and so the half-life of these molecules were incredibly short, and that was fine for cell lines, but when you start wanting to think about them as therapeutics, as Fernando's alluding to, you need to have an understanding about dosing frequency, and Ionis was certainly a pioneer. There's lots of other companies in the space today, but they were a pioneer in understanding how to modify the chemistries around the backbone to make antisense more drug-like, if you will.
Okay. And we talked a little bit about treatments for SMA and DMD. What do you think some of the benefits in pursuing ASOs over other potential treatments like small molecules, biologics? Why ASOs?
I'll start with that. So we're learning more and more that in diseases like ALS, you're actually probably looking at multiple diseases that have a very symptomatic manifestation, an actual pathological manifestation, where in this case motor neurons die. But we know that a lot of cases of ALS are caused by specific gene mutations. So at least 10% of cases of ALS are caused by gene mutations. So it gives us a little bit of a guidepost, knowing those gene mutations, that perhaps by going way upstream in the disease process at the actual gene and targeting that specifically, you could maybe have a profound impact for that limited set of patients. But it really kind of gives you like a smart bomb or a really targeted approach to ameliorate disease in these populations. Now, it still has these limitations where, these smart bombs won't work for somebody who doesn't have these mutations, but it's really incredibly promising for the people who do.
Yeah, I mean, when I think of that it's a really good question, actually. And again, I keep focusing on technology breakthroughs, but the other thing that happened along the same lines of developing this technology that really empowered us clinically to think about the utility of this platform was the sequencing of the human genome. When that draft sequence was developed 15 to 20 years ago, I think the first draft was published in 2000, there was this dream that if you could specifically target every single genetic mutation in the human genome, you could cure every disease. And when you think about ASOs, that is the best way to do it. If you can precisely get this technology platform delivered to the right cells, it could have profound effects on so many human genetic diseases.
Yeah, it sounds incredible. So where do you imagine the future for ASOs in, say, 10 to 20 years? We've already progressed so far in the last 20, so where do you think we'll be? It's a tough question.
I mean, we've already seen the utility of them in SMA is probably the prototypical example, or Duchenne muscular dystrophy, but those are really enabling the world, if you will. I think they're going to continue to improve in our ability to identify chemistries that are even better than the generations that we're using today. I think we're going to find better delivery mechanisms to make these technologies more drug-like, if you will. But there's other technologies that are competing along these lines as well. I mean, there's gene therapy technologies. Of course, we can't not mention CRISPR technologies at this point. Talons, zinc fingers. There's lots of technologies or gene editing tools that have clinical application. And I think right now ASOs are leading the way. And it's going to be interesting to see what happens over time.
I would say just one thing. A lot of the technologies that Steve just described, which are for gene editing, have some advantages over the ASO approach. ASOs are generally designed to suppress expression of a mutated gene, right? Sometimes the gene mutation, that means the new gene product, the mutated protein, is causing something problematic. But other times, the gene mutation is causing the gene to not work the right way, which means you're losing function. By suppressing expression, you may actually just be exacerbating that problem. So it's not like this approach can work for every gene mutation. It can work for gene mutations where you want to reduce expression. So these other technologies that Steve referred to have the potential to deal with both ends of that spectrum.
Okay. That sounds good. Thank you for your input today. That was really informative. For any listeners interested in this topic, ASOs will feature on future episodes of the Endpoints podcast, specifically those related to ALS. But thanks for listening and thanks for joining us.
Thank you, Emer. Appreciate it.
Thanks, Emer.
Thanks again for listening to The Endpoints. If you have any questions or feedback on this or any other episodes of the podcast, let us know by emailing endpoints@als.net.